Thanks for having me here. I actually cut a bunch of slides in this presentation, but I just decided to drag them back into the presentation last minute because we had some questions here. So about myself, this is one of the slides that I cut, but figured maybe I'd explain who I am. So I grew up in Kansas City, Missouri, and I went to school in Missouri. Also, I studied biochemistry, moved to Baltimore, studied at Johns Hopkins, and that's where I actually joined the lab with my former mentor, Jonathan Pesner. And we had a project around Sturge-Weber syndrome. At the time, we had some brain and skin samples generously donated by patients. And I'll talk a little bit about the work that we did to identify the mutation that causes the majority of Sturge-Weber cases. And so after I graduated from Hopkins, I stuck around a little bit into a fellowship, and I work now for Novartis. So Novartis is a pharma company. I actually work in oncology. So in my day job, I'm working on cancer research. And it says this year because it's an old slide, but I've been with the foundation volunteering as the chief scientific officer for about 2 years now. Okay, so just a really brief overview of the work that I did with Jonathan. We had, again, brain and skin tissue donated from patients with Sturge-Weber syndrome. 3 affected brain samples, tissue samples that were from hemispherectomy surgeries, and then 3 port wine stain samples. And in both cases, we had normal, sort of unaffected skin. Shouldn't say normal, but we had skin that was port wine stain, and then skin from right next to the port wine stain. And then for the brain tissue, we had brain tissue that had the vascular malformations and calcifications. And then we also had some more unaffected looking tissue. And if everyone kind of is familiar with the idea that we can sequence human genomes now, that wasn't like just a routine thing back in 2013 when we were doing this work. We had a genome sequencing center at Hopkins, and we had funding through the Sturge-Weber Foundation to sequence the genomes of these 3 Sturge-Weber patients, both the skin and the brain tissue. And why that was useful was that we could compare across all 3 patients what genetic mutations they all had and which they shared in common. And you could think about this as just like a little Venn diagram that I'm showing here. And this is actually the numbers of mutations that we found in each one of these patients. And you can see right in the middle, there's just 1 mutation that they all shared in common. And that happened to be a mutation in GNAQ. So we published this. I think it was really a lot of luck that we were able to have the right patient samples, the right funding, and the right timing to do that work. And since then, we've had a number of other groups confirm this finding. We also had access through the Maryland Brain Tissue Bank, 49 other patient tissue samples from Sturge-Weber syndrome, which we could then design sort of a validation to go specifically look at the GNAQ mutation in all of those samples and confirm that, yes, almost everyone had it. And there was a question earlier about whether, from Moth, I think, whether everyone that has Sturge-Weber syndrome has a somatic mutation in GNAQ. And the answer is probably not. So most people likely have a GNAQ R183Q mutation. There are some people that have that same GNAQ mutation at the same position, but it's a slightly different mutation. And there are others that have a mutation in a related or sort of sister gene called GNA11, which has more or less the same function. And then there's another gene that's sort of working with GNAQ and GNA11. And a few years ago, we actually had one of our research collaborators publish a paper describing a mutation in this gene as well. So all of these gene mutations are really highly related to each other. And so even if you don't have a mutation in the GNAQ gene, it's all caused by something that's very, very, very similar. And I think I mentioned that this was work that was possible because of really good timing and good luck. I think I can attribute a lot of our work to good timing and good luck. But the two other things were the tissue donations, which are really, really valuable. And we still really need to collectively organize and ensure that we have access to patient tissue samples when we can, because that enables new research. And the funding from the Sturge-Weber Foundation, which I'll talk a little bit more about now in my capacity as CSO. Thanks, Jeff, for introducing Courtney. So I also put together a slide just introducing Courtney as our scientific program director, and also just saying a little bit about what she's doing and hoping to do with the foundation. This last month was the first month that Courtney had started. So some of her responsibilities are developing a strategy to achieve the scientific goals of the foundation. She's identified a number of grants that we can apply for, both as a foundation and as co-applicants with our scientific collaborators. So really, funding is one thing that we're always looking for, both to drive the work that the foundation does to serve the patient community, but also to help build research programs to study Sturge-Weber syndrome. And Courtney's also really herself an excellent scientist. So she has biochemistry and molecular biology PhD, and will act as a liaison through the foundation around communications, answering questions about our scientific programs, and really helping to also organize our scientific collaborators with workshops and conferences each year. So she's fantastic. I wanted to give a little overview of what our research goals are in a couple different areas that I can talk about today. One thing that we'd really like to do is to develop what are called animal models of Sturge-Weber syndrome, and I'll explain that in the next slide. We want to organize research meetings, so we can't really drive the science forward unless we come together, ideally every year, with the researchers at universities who are studying Sturge-Weber syndrome, the clinicians that see patients with Sturge-Weber syndrome and also do clinical research, and talk about what we see and what questions we have and how we're going to answer those questions. And then related to that, we'd also like to try and bring in new people to study Sturge-Weber syndrome. I'm not that young. I'm on the young side, I think. And in general, we've got a lot of really great researchers, clinicians, and collaborators, but nobody stays in their job forever, right? And so we have to train the next new generation. And so the foundation hopes to establish things like travel grants to bring in young investigators. We'd also like to make sure that we're sharing our data and tissue as much as we can between our clinical and research sites. Again, the work that I did with Jonathan Pevsner, we wouldn't have been able to do that without the tissue sharing and the data sharing and the collaborative culture that we have. And the final thing is the foundation gives grants for researchers studying Sturge-Weber syndrome. These are seed grants. They're not huge amounts of money, but they can really get a lot of researchers started studying Sturge-Weber syndrome, especially if they don't already study it. And funding is really important to get people involved in the research. I work as an oncology researcher. There's a lot of money in oncology. People really donate to charities that are focused on curing cancer. And that while that's great, we really need to, bring that to rare disease and specifically to the Sturge-Weber research community. Okay, so I said I was going to talk about a couple of these areas that we, through the foundation, are trying to develop, right? So animal models. When when I say animal models, and when you think about like what is a model, we want to build something that's a research tool that you can use instead of a human, right? We can't do a whole lot of our research on humans. There are clinical trials that we do after you know that it's safe to give somebody a drug or some type of therapy, but you can't really ethically study humans in the way that you need to understand the basics about how something like Sturge-Weber syndrome develops. And so what we need are things like mouse models and zebrafish models. So the two pictures here. We actually have groups, Doug Marchuk's group, Joyce Bischoff's group, part of the Sturge-Weber Foundation International Research Network of collaborators. We have groups who have developed mouse models that allow us to study Sturge-Weber syndrome and how it develops in more detail. But there's still a lot of work that needs to be done to make these models useful and make them better. And so our research network is working on the next generation of these models. There's a lot of work that's involved there. Julia and now Courtney organize those research meetings every quarter, and we share updates, really generate a lot of excitement in the research community, because besides funding, the other thing that motivates researchers is being excited about what they're working on and being able to talk to each other and communicate that excitement is important. And so with these models of Sturge-Weber syndrome, we can answer questions. Like I saw in the chat, Wendy was asking if I could share some of the hypotheses about calcification in the brain. And, I think a little bit disappointed to say that we don't have papers and references that I can even share right now about, what causes the brain calcifications, because we don't really know that. But there are ideas that we have, and getting together and talking about them and figuring out which ones we can maybe test and how can we test them. That's something that the foundation encourages through our research group meetings. And so aside from these animal models, just getting together and talking about the research at a meeting is super important. And last year in July, we got together in Grapevine, Texas, for the SWFR meeting. So this is the research meeting that we hold. And this was actually concurrent with the family conference, which was fantastic. We got to meet with patients and families. And like Julia said, I think it was probably more interesting for the researchers to actually get to meet the families, because for a lot of us, that was the first time, especially for the younger people in the research meeting, it was the first time meeting somebody with Sturge-Weber syndrome, right? So that was great. And we used this as an opportunity to share research updates, figure out exactly how we can work together, and also what should our goals be to drive the research forward over the next 5 or so years. And I also wanted to talk today about another thing that the foundation has been encouraging, which is more data sharing. So the Sturge-Weber Foundation Patient Registry is a really fantastic resource, and all of you have contributed to this. And if you haven't, I would still encourage you to. This is a self-reported registry where you fill out survey questions, and these questions, the answers to these questions can help us to understand the patient journey. So like, what is Sturge-Weber syndrome like for you? Are there things that you share in common with other Sturge-Weber patients? And can we identify aspects of your Sturge-Weber that make you respond better to a certain type of treatment? And so what we've done this year is set up a collaboration with the Critical Path Institute out of Arizona. And here we're sharing our registry data. And the Critical Path Institute is also compiling other patient registry data from other rare disease foundations with a focus on rare epilepsies. So this is to really help potentially with accelerating future clinical trials for anti-epileptics. That would be something that we hope would come out of this. And the last place where the foundation really has a huge impact on research is its direct seed funding. So we have the Catalyst Award and the LISAS grant. And this last year, we were lucky to have two really strong applications. So we have one award for developing stem cell models of Sturge-Weber syndrome. And this is run out of Paracelsus Medical University in Austria. So we have international collaborators who are working on building, again, better models for studying the development of Sturge-Weber syndrome. And then we're also funding a study about how hormone changes in, especially in adolescence, might actually influence glaucoma development. So we don't know if there's a connection between your hormone levels and the severity of glaucoma or the development of glaucoma. But we hope to maybe test this by measuring intraocular pressure in people without Sturge-Weber syndrome. So actually, this hasn't been tested before, and it's just the very first step in understanding whether we can do a bigger study and whether there's something actually here. And what I'd like to do at the very end here is just say, you all can help out a lot with our Sturge-Weber research. I think the two biggest places are joining and staying in the Sturge-Weber registry. So this is still very important work that needs to be done. If you have trouble filling out the survey questions, let us know. And I think we can find a way to help with that. And if you haven't, we'd like you to find a way to enroll in the BVMC. And so that's the study that Dr. Loeb was just describing. And these two resources, I think, are both critical. We're doing this to try and help understand more about you and more about your journey, and hopefully do that so that we can develop therapeutics and better treatments to improve your quality of life.